The neurotransmitters are released from the axon terminal of another neuron into the synaptic cleft. As you can see, this is the axon terminal, and the neurotransmitters are being released. At the synaptic cleft, there are ligand-gated channels that help absorb and allow these neurotransmitters to be diffused through the membrane. When this happens, sodium channels open, causing sodium to rush into the cell. There are more sodium ions outside of the cell, and there are more potassium ions inside of the cell. The inside of the cell is more negative due to DNA. The outside of the cell is more positive due to the sodium ions. The sodium-potassium pump helps regulate the amount of sodium and potassium that are present both in and out of the cell at a given time or after an action potential. The rush of sodium ions make the resting membrane potential of negative 70 millivolts to become more positive. This is called depolarization. Once it reaches negative 55 millivolts, which is threshold, the sodium channels in the axon hillock, which is this area right here, will open up and a bunch of sodium ions will rush in. This will further bring up the membrane potential and depolarize it to positive 30 millivolts. When the sodium channels open up, the potassium channels also open up in the axon hillock, but at a much slower rate. Potassium leaves the axon hillock as sodium enters the axon hillock, but at a different rate. The sodium channels carry out the action potential in a domino effect, where one channel opens up and when the adjacent sodium channel reaches threshold. The action potential eventually reaches the axon terminal, where neurotransmitters in the axon terminal are released into the synaptic cleft. To go back to its resting membrane potential, the neuron uses its sodium-potassium pump to push sodium ions out and bring in potassium ions using ATP. Inhibitory stimuli, such as chlorine, might allow negative chlorine ions. So a chlorine channel might allow negative chlorine ions to enter the cell, which would hyperpolarize the cell, so it would make it more negative. So it would go to, for example, negative 80 millivolts. This can be seen through this graph. So right here would be negative 70, which is resting membrane potential, and then this would be threshold at negative 50, where we can see that action potential happening all the way up to positive 30 millivolts. So this part right here is depolarization. As soon as that action potential is reached, the cell starts repolarizing. So this would be basically when the from positive 30 millivolts, it would repolarize back to negative 70 millivolts using the sodium-potassium pump. However, if something like this happens over here with the chlorine ion, it would hyperpolarize all the way down to -80.